welcome to part 2 of chapter 4, cell structure chapter. Here we're moving on, learning more about different parts of the cell, and I've chosen to do it this way because I need to be able to point to specific structures with my pointer. So I made it really big here, and it's hard for me to do when I am in the video the way I normally am. So we'll see how this works. All right, so previously we talked about nucleus and ribosomes as two very important parts of the eukaryotic cell. Next is the discussion of the endomembrane system. So this is several membranes of different cellular organelles throughout the cytoplasm that work to divide the cell into several compartments. So membranes of different parts of the cell are said to be in this endomembrane system because those parts that are in the system have membranes that are either actually physically connected, like fused to one another, or they exchange parts of themselves with each other. And this will make a little bit more sense as we look at examples. So the members of the endomembrane system in the eukaryotic cell are the nuclear envelope. Remember, that's the double phospholipid bilayer, meaning there are actually 4 layers of phospholipids that surround the nucleus. That's called the nuclear envelope. The rough endoplasmic reticulum and the smooth endoplasmic reticulum, very similar organelles but have very different functions. Golgi apparatus, lysosomes, vesicles, vacuoles, and the plasma membrane. We've talked about the plasma membrane as the single phospholipid bilayer that surrounds the whole cell. So that's the thing that actually gives the borders of the eukaryotic cell. So let's look at this cartoony version drawing of the generic eukaryotic animal cell. Here is that nuclear envelope. So here it's like the light blue, but the outside of it is purple. And you see that there are 2 layers of membrane. As a reminder, when we say membrane in biology, that means 2 layers of phospholipid molecules that are organized tail to tail. We will talk more about membranes in the next chapter, but you do need to know at this point that a biological membrane is made up of phospholipid molecules that we learned about in chapter 3 that are organized in 2 layers where the long tails are facing the tails and the heads are facing inside and outside of that membrane. So each one of these light blue lines is representing that phospholipid bilayer, or those 2 layers of phospholipids. So there's one double layer of phospholipids here and another double layer of phospholipids here. Okay, we're now moving on from talking about the nuclear envelope to this next structure, this kind of like a maze of membrane, and this is called the rough endoplasmic reticulum, sometimes abbreviated as RER, rough endoplasmic reticulum. As it's trying to be shown here in this diagram, the nuclear envelope, the outside phospholipid membrane of the nuclear envelope, actually buds out and starts into making the rough ER. So they're actually connected, like physically, even though they're different colors here, they're not different. They are the same kind of phospholipid double layer as it was on the nuclear envelope. It just continues. It pinches out right here and continues. So those 2 membranes are completely connected. In fact, Maybe if there were aliens trying to come up with a way to name this, they would have named these two things the same thing. But the reason why we give them different names is because this light blue section, the rough ER, has a completely different function from the nuclear envelope, and it does have a somewhat different structure. So let's look at that. Endoplasmic reticulum, there is rough and smooth. The one we were just looking at that was light blue, maze-like, that is connected to the nuclear envelope is called rough endoplasmic reticulum. The reason why it's called rough ER is because it has a lot of ribosomes, the little protein makers, parked onto the surface of the rough ER. So when scientists were originally looking under the microscope, it looked like it had like little studs on it. So it looked rough, as if it would be rough to the touch. Okay, so it's not actually rough to the touch because everything there is pretty squishy, but it would look like that from a stained microscope slide. So we call it the rough ER. So rough ER is this blue, light blue. It's not actually blue. In this picture, it's blue. It's studded with the lots and lots of ribosomes, the protein maker machines. As you can see them, these tan dots. Notice there are actually also ribosomes on the outer part of the nuclear envelope as well. Okay. The main function of rough ER is to synthesize proteins, and specifically it's to synthesize proteins that are to be secreted. Okay, so we learned that it is the actual ribosomes, those little tan studs, that build the proteins. That's correct. So if I were to ask you what builds the proteins, you should say it is a ribosome. However, if that protein is going to be secreted out of the cell, so it's made for a neighboring cell in the multicellular body, or for a cell somewhere far away in a multicellular body, so the protein has to be secreted like it does out of our pancreas, then the proteins that are made by the ribosomes go into one of these pockets from the ribosome into one of these inside areas, which are called cisternae, and there they are finished being created. They're modified and they're folded and they're packaged to be secreted out of the cell. The smooth endoplasmic reticulum, or the smooth ER, has a wider variety of functions. What you need to know about it for now is that this is where the cell makes their lipids, so this will include steroids that are necessary for making hormones, right? As we learned about in the previous chapter. Smooth ER also stores calcium ions. This is important for muscle cells that require calcium ions in order to contract. And it's important for detoxification. So what that means is that there are proteins in the smooth ER that can detect toxins in the cell, and they grab that toxic molecule and they do something to it, often sticking a hydroxyl group in order to make it less toxic. So sticking a hydroxyl group makes a molecule more water soluble, which means it is easier to flush it out of the body. Okay, there's a lot of smooth ER in the liver cells because liver has many functions. One of them is to detoxify your body, right? So people who abuse alcohol have livers that are unhealthy because they end up being overworked for this function of detoxification. The ratio of smooth and rough ER depends on the cell's function. So cells that are tasked with synthesis of proteins that are to be secreted will, like pancreatic cells, have a large ratio of rough ER. They just have a ton of it. Whereas cells that synthesize lipids or detoxify, they have more smooth ER. So that would be liver cells or testes and ovaries, which make hormones, and those are sex hormones, are made out of steroids, so they are lipids. Okay. Notice here that the membrane that makes the rough ER just continues on and buds out to become the smooth ER. Remember again that when I say membrane here, it is a phospholipid bilayer, and that is like a little fat bubble that has space on the inside, because phospholipids are lipids. So this is not an exact analogy, but think of a little oil bubble that is on the top of your soup, and then there's a bigger oil bubble around it. They can connect, right, with like a skinny part in between them. And that's how the phospholipids are connected. Of course, these structures are more stable than the random fat bubbles floating on top of your soup. So they just don't just like constantly change their shape, but they are flexible and they can change their shape. Your cells can continually build more or less rough ER or smooth ER as is necessary. Okay, so you need to know that both the smooth ER and the rough ER are made out of the membrane, which is itself phospholipid bilayer. The rough ER has a lot of ribosomes parked on it to create proteins that are then pushed inside. There are also a bunch of proteins inside that will finish up the job of creating new proteins. Whereas the smooth ER does not have ribosomes, so it appears to have a smooth shape. And it is made up of a phospholipid membrane, and the inside has many proteins that are going to do jobs of creating lipids and detoxifying. Okay. Here is a colored microscope image that is trying to show you what it would look like if you looked at the smooth and rough ER of an animal cell under a light microscope. Continuing on with the endomembrane system, so the membranes being connected or interacting, is the Golgi apparatus. Golgi apparatus is another organelle inside of eukaryotic cells, and it is also made mostly out of phospholipids because it's part of the endomembrane system, meaning most of it is just membrane. When we say endomembrane system, endo means within or inside. So it's the system of membranes that's inside of a cell. The Golgi apparatus kind of looks like a stack of pita bread of various sizes. And those stacks are called Golgi bodies. I'm not going to ask you about the Golgi bodies, but you do need to know the general structure of Golgi apparatus. Okay? And let's look at what that looks like. So this is a cross section, right? They're not normally open pita bread like this. They would be closed on both sides. It's these stacks. And again, all of this is made out of phospholipid, is made out of phospholipid bilayer. So it's very gooey, very flexible, kind of like a bubble of oil on top of your soup. There are two distinct sides of the Golgi apparatus. There's the side that faces closer to the nucleus and the endoplasmic reticulum, and it's called the cis side. and the trans side is the one that faces the plasma membrane. Now, the main function of Golgi apparatus is to prepare things that are going to be secreted out of the cell. So proteins or lipids that are going to be secreted outside of the cell. It packages them and puts little labels on them as if to tell the outside of the cell world where this package is supposed to go. And the way that it works is really cool, and I'm going to show you here. So again, this is a cross section so we can see what's going on the inside. Light blue right here is the rough ER. So in here, proteins are synthesized to be secreted out of the cell. So let's say it's insulin protein that is being made in the pancreas. So it was probably put together on one of these ribosomes, this tiny tan or brown circle thingy that was parked on the rough ER. But then as the protein was built out of the amino acids, it got shoved into the cisternae or into this middle part of the rough ER, where the proteins that are already hanging out in there finish creating the correct 3D structure of the protein that we're making, insulin, and it's shoving a bunch of copies of that insulin to the side. Because this right here is made out of very flexible phospholipids, like a fat bubble, it can just bud out. Okay, so imagine you have a giant bubble of oil on top of your soup in a bowl floating around. You can actually experiment with this if you have fatty soup sometime soon. You can grab like a spoon or a fork and just kind of pull on the edge of that bubble. And what are you going to get? You're going to get one tiny bubble of fat separating from the bigger bubble. Can you imagine that happening? If not, you can maybe try to experiment with it. It works well with like creamy soups, so you can see the fat floating on top. Anyway, so once the proteins are finished being synthesized inside of the rough ER, the fatty phospholipids bud off and create a transport vesicle, which is it's like a tiny fat bubble, but with space inside for things. It's like a little package. Then it is carried. It looks like it's floating, but it's actually carried. We'll see how later. And then it goes to the cis side or cis face of the Golgi, and then it bloop and fuses with the fatty phospholipid membrane of the Golgi. So here it would be like a tiny fat bubble on the surface of your soup being pushed toward a bigger bubble, and then it just fuses and becomes part of that bigger bubble. Can you imagine that? So now this phospholipid part fused, and the way it fuses, the proteins that were inside of that sphere, inside of that vesicle, get dumped into the cisternae or the interior pockets of the Golgi apparatus. Here they will go through that tunnel and all those stacks. They will get little molecules stuck onto those proteins. So a lot of times it'll be like a phosphate group will get attached, and that will work as a male tag for this stuff. Then it travels to the trans side of the Golgi, and again it'll bud off, little fatty bubble buds off with your ready to go insulin that is ready to function outside of the cell. That vesicle is now called secretory vesicle because it's to be secreted. And it goes and it bloop, and it fuses with the phospholipid membrane of the cell. So this was the plasma membrane or the outside border of the cell. And your phospholipid vesicle fused with it in a way that dumps the contents to the outside. So a phospholipid molecule that used to be right here and then spent some time being right here is now right there. So there's a constant exchange of phospholipid, the membrane building molecules, between these different parts, which is why we group these organelles into a club that we call the endomembrane system, right? So it's attached to the nuclear membrane and attached to the smooth ER, and then vesicles or little transport spheres from the smooth ER or from the rough ER can go to the Golgi. So if you were a lipid made in the smooth, you can go to the Golgi, or protein made in the smooth, you go to the Golgi, where it packages it for secretion, and then it blops out a bubble full of the product, which fuses with the plasma membrane and dumps the product to the outside of the cell, which could be the bloodstream. So now your insulin is out there, out of the cell, in a bloodstream, doing the thing that insulin does in our bodies. Okay, so I hope it makes sense how all of these membranes work together. I will mention one more thing about the Golgi, and that is, in addition to being like the packaging facility, like a FedEx, of products that are made in rough and smooth ER, it actually also synthesizes carbohydrates for the cell, and high carbohydrates that are to be secreted. So that's a little side note that's especially important in plants. Lysosomes are also part of the endomembrane system because they are like a unique one of those bubbles that we saw. Instead of transferring things from a place to place, it has a different job, which is to break down or digest and recycle things that are not needed in the cell anymore. Okay. The lysosome is made by the Golgi, so it buds just like this, it buds from the Golgi, but it doesn't work to go out and secrete stuff. What it does is it has a lot of acid inside of it, and it has special proteins that are like little choppers in there. You can use it like an incinerator. Let's say this is an organelle that we haven't learned yet, that is too old and it doesn't do a good job in the cell anymore. It needs to be gotten rid of. So the lysosome, again, this is a cross section. Normally it's a sphere. It engulfs, gets itself kind of around this large thing, and it can do it because it's stretchy and it can be large. And the acid, as well as the chopping proteins, will take this organelle and break it into pieces. And then those pieces will be spewed out and can be reused by the cell for making new stuff. And this is one of the kind of extreme examples of how having these compartments is like that example of having an oven where you can bake your cookies, whereas if you didn't have an oven, you couldn't just heat up your whole house to that hot temperature. So here, the interior of the lysosome is dangerous because it breaks things down. It like deteriorates things. It incinerates things with its acid and chomping proteins. So you don't want that stuff on the inside to just be, all around your cell. It'll just start chomping everything down that it's useful to the cell. So it can have all the digestive stuff inside safely and in a very controlled manner. For some cells, that they can actually sense something outside of its plasma membrane. So if a cell senses something on the outside of it, it can engulf it by just making a little phospholipid vesicle, which means like a sphere of the phospholipids, by having some of this fatty stuff just pinch off, and that stuff can be digested by a lysosome, just the same way as this is. This is actually how amoebas and lots of other single-celled eukaryotic organisms eat. You just engulf little organic particles and then you break them apart into small monomers, and then you spew those out and you can use them to do work inside of your own cell. But even in our bodies, this system is useful. Our one kind of white blood cells in our body kind of crawls around the blood, and it can sense things that are not supposed to be there. Debris, bacteria, whatever is not supposed to be in your bloodstream, and it can slurp it into a bubble by pinching its own plasma membrane around it. And then that vesicle will be fused with a lysosome, where whatever that stuff was will get chomped into its monomers, which are then spewed out and reused by the cell. So that's lysosome. There are other microbodies, so similar working spheres that are made out of phospholipid membrane, double layer, to make that bubble or that sphere. And then inside it has various different proteins. So lysosome has chomping proteins and acid, whereas peroxisomes, they have enzymes, which is a kind of proteins that are to break down fatty acids. So it's for breaking down of certain things. Okay. You don't need to know a lot of details about it, but we will mention peroxisomes in a future chapter, so I just wanted to introduce you to it. Vacuoles are very similar to vesicles. So remember vesicles? All these little transport vesicle, secretory vesicles, so the thing that's about to be secreted. So vesicles are often small, they fuse with other membranes, and they're like a transition sphere made out of phospholipids. Whereas a vacuole is a permanent bubble with various functions. Our cells do not have vacuoles, but plants and protists, so other eukaryotic organisms that are not fungi or animals, have them as well. So for example, here is a vacuole in a plant cell. They are usually super huge. If you look at a plant cell under microscope, it usually looks like it's completely blank for most of it on the inside, like it looks just empty. The reason why it looks like that under a simple light microscope is because you have this giant vacuole. So the edges of it are made out of phospholipid bilayer. It's a giant three-dimensional blob. And it's used for storage of different things. And in a central vacuole is generally for storing water in a plant cell. That's why you can't see it in the light microscope, because light shines straight through it and just looks like it's empty. Okay, but vacuoles are used for storage. They're used like for a bunch of water that single-cell organisms can squeeze out and use that to propel themselves to move around watery environments. So that's pretty cool. You will see some of those in lab at a later time. Okay, that was it for the endomembrane system. Remember, endomembrane is internal membrane system of the cell, includes all of these structures because all of their membranes or all of their borders are made out of phospholipid bilayer, so double layer phospholipids, tail to tail, in a way that is either connected, like nuclear envelope to rough endoplasmic and to smooth endoplasmic, or they bud off, like pinch off of one another and exchange parts of the membrane with one another, like in rough, smooth, endoplasmic reticulum, Golgi, lysosome, vesicle, vacuoles, plasma membrane. Sorry, vacuoles do not exchange, but the others do. Okay, That's it for the endomembrane system. And now we're switching gears to talk about parts of the cell that are involved in energy handling. Mitochondria are found in all eukaryotic cells. So protists, animals, plants, and fungi all have mitochondria. Most of our cells have several mitochondria or even many mitochondria. Mitochondria can come in various shapes, but we generally show them as like a sausage or oval. They can be also round. They are bound by two double phospholipid bilayers or two membranes, and they process most of the energy in our cells. Let's look at their structure first. Okay, here is a cross-section of mitochondrion. So a single is mitochondrion, plural mitochondria. Okay, so this right here is representing a phospholipid bilayer. So one biological membrane is just like a big bubble, like we have seen in vesicles and vacuoles and the plasma membrane. Very similar. On the inside, you have another spherical-ish membrane, but it's shoved in such a way that it creates a lot of folds, and we call those folds crista. One fold, plural, is cristae. You can imagine it's like you have a small Ziploc bag with a giant garbage bag inside. So it had to be all folded and has a bunch of folding in areas on itself. Something cool about mitochondria is that they have their own DNA. And this might not seem weird to you because you don't know a lot about DNA yet, but actually super, super weird. And one of the coolest things in biology to me, I mean, okay, there's so many cool things. How do we even pick? All right, let's back on topic. Has their own DNA, and they have their own ribosomes that are different than the other ribosomes that live elsewhere in the cell. I will tell you why that is in a second and why it's weird. But before we go there, you need to know that there's an outer membrane. There's an inner membrane that's folded. The space between the two membranes is called the intermembrane space. And the space that's inside the inner membrane is called the matrix. Okay? The reason why it's important is because mitochondria process the energy that we get into our cells, usually in the form of sugars. So if you have a glucose, you cannot use the glucose to do any work or anything useful in your cell by itself. You have to convert the energy that is stored in the bonds that are in the electrons of the glucose into a useful form. Mitochondria do this. It's very cool, and we will spend a whole half of a chapter talking about mitochondria in just a couple of chapters. Okay, that's it for energy processing organelle inside of animal cells. Again, these are also present in fungi, protists, and plants, but plants have both mitochondria and chloroplasts. I'm going to say it one more time so you write this down and so you can answer this on the test. Plants have both mitochondria, the plural of this thing, and chloroplasts, both unique organelles that are important for energy processing. Chloroplasts, something you probably have heard of, they are organelles that are responsible for photosynthesis. They contain a molecule called chlorophyll, which is why chloroplasts are green, which is why most plants are green. And they, with the help of chlorophyll, chloroplasts are able to capture the energy from the sun and make glucose out of this energy. I will say that one more time. They are able to capture chloroplasts with the help of chlorophyll, are able to capture energy from the sun and incorporate that energy into a glucose molecule, which is made out of carbon dioxide. Okay. So carbon dioxide plus energy from the sun is what chloroplasts use to create glucose. This is what they look like. They also have 2 phospholipid bilayers. So again, an outer membrane and inner membrane. They have these little stacks that are called thylakoid, granum with thylakoid membrane around them. They have their own ribosomes and their own DNA. It's also weird. It's probably not sounding weird to you, but it's super weird. I'm not going to ask you a lot of questions about the chloroplasts because we are focusing on you going into the health professions for humans or other animals, and therefore you don't need to know a lot of details about photosynthesis. But you should know what the function of a chloroplast is and the fact that it has 2 membranes, has its own DNA and its own ribosomes. And here's why both of those things are weird. The leading scientific theory about mitochondria and chloroplasts is called endosymbiosis theory. And it proposes that way back in evolution, there was a bigger cell that engulfed, basically like ate in one bite, a protobacterium, meaning like an ancient type of bacteria-like organism. And instead of digesting it inside, it just kept it whole and kept it alive inside. This little protobacterium that's now inside the cell was able to process energy, which helped the bigger cell. And the littler engulfed prisoner also benefited because he was getting totally steady supply of food, shelter from predators, etc. So you just have this cush life, but you're providing the energy molecules that you process for the bigger cell. And they lived together for so long, meaning every time this cell had to reproduce, it also reproduced this inside. And so there are many millions of generations of this, to the point where they become so codependent they cannot exist without one another. And another one of those engulfing situations happened when a cyanobacterium, so a photosynthesizing bacterium, was engulfed. This happened to plants. It did not happen in animals. And the reason why it's pretty certain that these guys, chloroplasts and mitochondria, once upon a time, billion or more years ago, was its own individual, separate bacteria-like organism, is because the DNA that's inside of both of these organelles now is extremely similar to the DNA of all bacteria that exist today. Like, it looks the same as bacterial DNA. And the ribosomes in there, they're not the type of ribosomes that our cells have on the rough ER or on the outer surface of the nuclear membrane, or the ones that are free floaters. The ribosomes are like the ribosomes in modern day bacteria. So the whole thing is really cool. There's a lot of research about it to support it, and it's one of the most fascinating things to me about cellular biology. Okay. I'm going to ask you very briefly, what is the endosymbiotic theory or endosymbiosis? It's that today's eukaryotic organelles, mitochondria and chloroplasts, evolved by symbiosis. Sim is together, bio is to life, sys is the process. So process of living together arose from two or more cells that used to be separate but ended up together for so long that they're inseparable now and cannot survive. Mitochondria cannot survive outside of a cell anymore. Neither can chloroplasts. Okay, so that is it for the energy producing organelles, mitochondria, and chloroplasts. And I should point out that this is a common sentence, energy producing, but it is actually not accurate. I should say energy processing organelles, mitochondria, and chloroplast. Energy processing organelles, mitochondria, and chloroplast. One of them, chloroplasts, is able to capture energy from sunlight and therefore build glucose molecules out of sunlight energy and carbon dioxide, while mitochondria are able to take glucose and break it down in a way that makes it into useful energy for the cell, a process we will talk about in details later. Okay, we're going to move on to the cytoskeleton. Remember, cyto means cell, skeleton is the skeleton of the cell. Not made out of bones, but it's made out of fibrous proteins. What does it mean when you say fibrous proteins? It means a ton of proteins stuck together to create a long fiber-like structure. So just like a thread-looking thing made out of proteins. So a cytoskeleton is a network of protein fibers. Think of it like a bunch of really strong ropes that are found in all eukaryotic cells in order to support the shape of the cell, keep the organelle in fixed locations, and it's also useful for transport. I will show you in a second. The system is extremely dynamic. So it's not like our skeleton that's more or less fixed. These ropes, so to speak, of protein that make up the cytoskeleton are constantly being built in one place and taken apart in another place, and elongated on this side and shortened on the opposite side, constantly changing. There are 3 types of fibers. So 3 long types of protein structures: microfilaments, microtubules, and intermediate filaments. I'm going to show you the diagrams and tell you about them. You can go back to the slide after pausing if you want to write down some of these notes. Okay, so this is showing us a portion of a cell. This at the bottom is representing the plasma membrane, which is also called cell membrane sometimes. Here, what's this blue thing? Come on, we just talked about it. Yes, rough ER, because it has ribosomes stuck on it, and it's like these flattened structures, tubey structures. Okay, here's a mitochondrion, cross section of this, and these fibers, which is, when you say fiber, it means it's like rope shape. So anything that's fibrous, it means it's long and skinny shape. There are 3 of them. The first one is called a microfilament, and it's like 2 pearl necklaces twisted around each other. Each one of these circles is a tubulin protein. So the circle is 1 protein, 2nd protein, 3rd, but they're identical. All these proteins are, well, there's really 2 types of tubulin, but you don't need to worry about that. There's 2 types of tubulin protein that are structured. Oh crap, I just messed you up. Tubulin is over here because I'm not talking about tubulin. I'm talking about filament. So yes, I was right. I'm sorry I can't stop this and go back because of the pointer thing. Maybe I can. Okay, I'll try to cut this out. But each one of these is identical. They're called actin proteins. This is an actin protein. This is an actin protein. This is an actin protein. So millions of actin proteins connected in this shape. Okay, so these are the skinniest of the 3 types of cytoskeletal structures. The fattest one is called a microtubule because it's a tube. It's hollow on the inside. Each one of these circles is representing a different protein. The dark green ones are all one type of tubulin, and the other is a different type of tubulin proteins. Okay, so alpha and beta tubulin. All you need to know, it's made out of tubulin proteins that are structured to create a tube. Okay. And the third kind is actually called intermediate filaments. They're mostly present in animals only and only in some cells. And they're really strong and they're a lot less dynamic than the other two. So these guys are almost permanent. It's hard to take them apart, lengthen them, shorten them. All of that is hard to do. Whereas both of the other microtubules and microfilaments or actin filaments, 2 names for the same thing, actin filaments or microfilaments, are very dynamic, easy to take apart and put back together. We will come back to talking a little bit about these at a later time. Centrosomes are made out of microtubules and they organize microtubules. Hard to explain at this point because we have not learned anything about any of this stuff, but I'm going to introduce you to the fact that they are microtubule organizing center. And most likely will not test you on this, but I want you to be familiar with the term so when we revisit it in chapter 9, I believe, you are not hearing it for the first time. So associate centrosomes with these green tubes, okay? Now, for cell movement, organelles are necessary. And I should say, for cell movement, cytoskeletal structures are necessary. So essentially, all cell motion is tied to actin, microtubules, or both of these filaments. Okay, so again, it's actin filaments, which are also called microfilaments or microtubules. They're extremely important for cell motion. Some cells crawl, like your white blood cells actually crawl through your blood vessels. That's using the constant lengthening and shortening of microfilaments, or those actin filaments. The one that looks like 2 pearl necklaces wound around each other. And the way it does it is the cell is pushed by microfilaments growing on one end and shortening on the other end. So think of like a gerbil in a wheel or a hamster inside of a wheel, and it pushes on one side and takes pressure away from the back, and that makes the ball go forward, right? So these guys aren't pushing in that same way, but they are building it longer on the front, which then pushes. Does that make sense? Okay. Flagella and cilia. We saw flagella in the picture of, it's like a long tail on that bacteria at the beginning of this chapter, just usually a one long tail. In human cells, there's a flagellum, which is a singular for flagella, that's obvious on only one cell type, and that's the sperm. But we used to have flagella on many more cells, and you can see the remnants of that in several places. So you don't need to remember that, but flagella are super important for motion of sperm, and they're really important for motion of many other organism types that live in watery environments. Cilia are like little hairs on the outside of the cell, but they're not actually something stuck to the cell. They are the protrusion of cytoskeleton from inside. Okay, this is how I'm gonna have you imagine that for both flagella and cilia. Okay, imagine you have a balloon, which the latex of the balloon is going to represent the phospholipid bilayer or the plasma membrane of the cell, right? The outside barrier of the cell. And you're going to pretend like your hand is inside the balloon, and then you stuck a finger up pressure toward the latex. What's going to happen? The latex is going to like surround your finger and it's going to be protruding, but the latex is going to be covering your entire finger, right? So in that case, the finger is going to be microtubule or microfilament, those cytoskeletal structures, while the latex is representing the plasma membrane of the cell. And this is the same similar thing from flagella and cilia. It's just that the finger part or the cytoskeleton part is different in both of them, and it results in a different shape that sticks out. Okay, so here, this is the surface of the balloon, and this is your finger sticking out. So you have the plasma membrane surrounding it, and on the inside, you have the structure of a bunch of microtubules. Okay, so I just want you to know that flagella and cilia are made out of microtubules that are in special arrangements sticking out of the plasma membrane. Okay. That's it for motion. I want to mention a couple of things that are on the outside of the cells. So we call that extracellular structures on the outside of the plasma membrane. Many eukaryotic cells, as well as pretty much all prokaryotic cells, have cell walls. This is not the same as plasma membrane. This is an actual like hard, non-flexible structure that is surrounding the plasma membrane. So here, this tan border is the phospholipid bilayer that is the border of the cell. It's flexible. Again, think of the cell as a water balloon, and the latex of the water balloon that's squishy and flexible and stretchy is the plasma membrane. Now this thick green stuff, it's not normally green, it just looks green in this picture, is made out of a bunch of different carbohydrates mostly that surrounds it. It's a hard structure. It's not flexible. You can't just like push on it and have it easily give. That's a cell wall. Okay. There's a lot of complexity to it, and it differs between fungi. Remember, in fungi, cell wall is made out of chitin, that carbohydrate. In plants, it's made out of a different carbohydrate. Prokaryotes, so bacteria and archaea, have a different cell wall. Okay. Plants have 2 cell walls. You don't need to worry about the details, but you should know which kinds of organisms have cell walls. Plants, fungi, protists, and prokaryotes, which is bacteria and archaea, have cell walls made out of various carbohydrates. In fungi, they're made out of chitin. In plants and many protists, made out of cellulose, both carbohydrates. Okay. Now, I used to think when I was, first starting college and not taking a lot of biology classes, that our body, us as in animals, has just like cell next to a cell next to a cell really, really tightly. And that's like what we're made out of, because I heard we are made out of cells. However, that's actually not true. Depending on the tissue type, there may actually be a lot of space between cells. Yes, in some places it's cell next to a cell next to a cell really tight, but in most places it's actually various amount of space between cells. And that space is called extracellular matrix. Again, extracellular means outside of the cell. And the reason why this extracellular matrix is important is because we do not have cell walls and cannot protect our cells on the outside very well. So our cells secrete a bunch of goo, and it's watery, made with a lot of glycoproteins, so proteins with sugars attached. And it makes like a jelly, gooey substance in between our cells. There are also a lot of different proteins that are connecting and holding things together. It protects the cells, their surface. It helps cells communicate with one another. It is extremely important, and that's all you need to know for right now, okay? Space between cells is called extracellular matrix. It's present in animal cells only because fungi and plants have cell walls, so they don't need this. And other organisms are single-celled organisms, so the space outside of their cell is just like the world. But for animals, we're still all inside the body, but the space between the cells needs to be controlled and protected. It's called extracellular matrix, and very important. Here I'm going to show you a little cartoony version. So this is the plasma membrane, one layer of phospholipids and a second layer of phospholipids, these tails facing tails that are hydrophobic. The heads are facing inside the cell and outside the cell. This is the cytoplasm, which is the inside of the cell. You have these large proteins going through the whole plasma membrane and attaching to the actin filaments, also called microfilaments, of the cytoskeleton. And then on the outside here, it's attached to a bunch of other fibrous proteins, meaning proteins that hook together to form long thread-like structures. In this case, it's showing collagen. There's elastin. You might have heard of that for like, ooh, buy this lotion. It's going to help you with the elasticity of your skin. And various other things. And it's a watery, gooey, protein-rich environment. Okay. This is comparing some of the structures that we've talked about, prokaryotes, right? Prokaryotic cell types are for bacteria and archaea. They do not have a nucleus and lack membrane-bound organelles. Then there's animal cells, plant cells, and we don't have fungi here. Okay, so make sure you just briefly review this about the structures that we've talked about. The last thing I'm going to mention very briefly here is cell-to-cell interactions. So sometimes cells need to be really well connected or communicating with neighboring cells. And there are 3 different ways that they can do this. Actually more, but we're exploring 3. For cells making contact with one another, recognizing each other, etc. This is important. 3 categories are discussed in your textbook. The reason why I am mentioning them here is because when you begin anatomy and physiology, one of the first things that you learn are tissues, which are made by connections of various kinds of cells. And the way they're connected is using these things. So you need to be familiar with them, even if we're not going into depth here in this class. Tight junctions are when proteins are used to connect plasma membranes of neighboring cells. So here, tight junction. It's like every one of these studs, it's like a protein that is as if the two plasma membranes are being sewn together. So this is an edge of one cell, and this is an edge of a different cell. So there's a whole cell here and a whole cell here. You want to connect them to be really, really tight sometimes, so there's no space in between. And imagine like your sewing machine and you're going and quilting in between to keep them tight. This is especially important for tissues that need to form tight sheets like your skin. Okay. The second is anchoring junction. It's where a cytoskeleton of one cell attaches mechanically to neighboring cells. So let's look at an anchoring junction. Here are two cells, and you don't want to sew the whole membrane together, but it's like a Velcro in between. And this is one of the cytoskeleton structures, like microtubule or microfilament, that goes through here and in between and into the other side. Okay. And lastly is communication junction. And this is actually more of a tunnel than a gluing things together. So when you want to have a chemical or electrical signal go from one cell to another. So that means you want to actually be able to move electrons from one cell to another, or ions or other chemical entities. Okay, so here you see it's like a tunnel that goes from one cell to another cell to easily bulk flow substances through. And that's it for our tour of the cell. I know there's a lot. Create yourselves tables and charts so you can keep this information straight. I wish you luck and talk to you soon.